Hi guys, I welcome you all to the next lecture in our anthropology series. So today we are going to start with our next topic, that is 9.4, wherein we are going to so 9.4 we are going to study about chromosomal aberrations, chromosomes and chromosomal aberrations in man. Herein we are going to cover different kind of diseases that are there because of genetical mutations or chromosomal aberrations. Okay, so okay, I want to take it very slowly because there are a lot of new words and terms that you might not be aware of. So I told you also that I am going to cover each and everything in utmost detail, so do not worry about it. Let's start with it. So now I talked about mutation, right? So it is a sudden, or a you can say sudden change in the normal gene or gene frequency, right? So in this what happens is that there are two kind of changes that can happen to a chromosome. Now just try and understand. Look at this picture. Okay, first I'll give you a brief introduction of what chromosome is and all of that, because I told you. Okay, so one chromosome, ठीक you can say looks somewhat like this. Okay, this is one chromosome, ठीक and in every organisms these chromosomes have the DNA which encodes all the genetic material in a living organism, and these together these chromosomes or different set of chromosomes are called the karyotype. Okay, now let me just show you the picture. What is a karyotype? I'll just show you. This is a human karyotype. Okay, why this I'm calling this as a human karyotype? Because for every organism it is different. For example, some organisms might not have 23 pairs of chromosomes but have like 30 pair of chromosomes or 12 pair of chromosomes, like that. So it differs for each organism, right? But in humans case, you know that we have 22 autosomal pairs. Till here we have the from 1 to 22 we have the uh, no, autosomal pairs. Okay, and then we have either an XX or an XY, which is the sex chromosome, right? So these are the 23 set of chromosomes that are there, and in a human being it looks like something like this. So you can see it. Okay, now what happens is, I told you this is a chromosome, I mean this is a pair of chromosome. When now what happens, for example, this is a pair of a chromosome of the father, and this is let's take as this is the mother's chromosome. ठीक है, so now they are अभी के लिए they are both diploid. That means they have two pairs, right? They have two parts, right? So these are diploid. So at the time of, you can say, meiosis, right? It becomes haploid. That means single single. It bifurcates into single single. Let me just show you that also. It will become like this. It will become like this, like this. It will break like this. No, it will break like this, and the green one will also break like this. It will break, right? Sorry, it should go in this direction, like that. So they break like that, and then they join in the next uh, you know in the next generation. That means the child, because both of them will contribute one part of it, so it becomes the, again it comes in the diploid form in the next generation. Okay, so this is how the recombinations of both parents, uh, chromosome or DNA, you can say, happens. Okay, so this is what it is. And now there are different types of chromosomes. Okay, not all, as you can, I'll just take you back to this picture. You can see not all of them look the same. Some are longer. For example, this one is the longest, this one is the longest, then these are very short, and then some are different. For example, XY. X is a long, you know, uh, 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 pair. In the pair, X is long and Y is short, right? So from here you can see karyotype में हम क्या करते हैं, उनको डिसेंडिंग ऑर्डर में लगाते हैं, राइट? तो हमने देखा यहाँ पे, हमने बेस्ड ऑन इट्स हाइट हमने कुछ डिसेंडिंग ऑर्डर या कुछ एक सिस्टमैटिक वे में वी हैव ट्राई टू अरेंज देम, एंड एक्स और एक्स वाई वाला लास्ट में अरेंज करते हैं, राइट? So this is one thing. Then they have differences in their, even in their shapes and even in their sizes. So this is a little bit that you should know before you start to study about chromosomes. Now I'll just tell you different types of chromosomes. For example, this one, you can call this one as a submetacentric. Okay, the first one you will call as a submetacentric. I'll tell you what is it, don't worry, just try and understand. Okay, let me find some other which from which I can explain you. Okay, okay, let me just draw it only here. You've seen the pictures, right? So let me make it for you. Okay, this is one type I am making. This is type number 1. Then type number 2. This is type number 2. Then comes okay, and this is type number 3, and we have ठीक है type number 4. Okay, so this one, the number 2 one, shows that, okay, now just understand the parts of a chromosome also. जो the upper ones we will call the P arm, ठीक है, and the lower one we'll call the Q arm of a chromosome. So you need to understand, because आगे हम थोड़ा technical भी use करने वाले P arm and Q arm. So upper ones are called P arm, नीचे वाला the bottom ones are called the Q arm. Now just try and understand what I'm trying to say. In here, the second one, the P arm and the Q arms look equal in size, right? So that is why they are called metacentric. They are called metacentric because this is the center or the centromere of a chromosome. It is a technical name, centromere. From there, both the P arm and Q arms exactly, you can say they look not exactly, but they look equal, right? So it is called metacentric. The second one, in that P arm थोड़ा सा Q arm से little bit shorter है, ठीक है, that is why it is called submetacentric. Submetacentric. Then comes your telocentric, which is the third one, जिसमें you don't find the, or it is the P arms very small or not even there, ठीक है, that is telocentric. And the fourth one is called the acrocentric. Okay, इसमें भी P arm is shorter but very short, मतलब shorter than even the submetacentric, ठीक है. So you can see the proportions here, ठीक है. So like that, so you understood different kind of shapes of chromosome, ठीक है, तो आप ये समझ गए होंगे. So based on this, now we have two kind of because now we are trying to study chromosomal aberrations, ठीक है. Now what is chromosomal aberration? That means whenever there will be any shift from the usual type of chromosomes, ठीक है, in a human being, it will be called as an aberration. Okay, so normally we have 23 pairs. That means we have 46 chromosomes. Herein we have 22 autosomal pairs, and we have one sex chromosome का pair, right? So this is the numerical way, ठीक है. And then we have this normal karyotype of a human being looks like this. That means chromosome are there in the pairs, ठीक है, and they something look like this, right? So there are two kind of aberrations that can happen. One is in the structure of the chromosome, which is इसमें in this karyotype any structure of the chromosome changes, that can be one kind of aberration. The second aberration would be in the numerical number, either anything in the number of the chromosomes change, that is going to be another kind of chromosomal aberration. So now you understand what we are going to study. So what is a chromosomal mutation? That is either change in the structure of the chromosome or the number of chromosome. Okay, number or structure may change. Okay, let me change the color of the pen. So in this we have types. We have heteroploidy or genomatic mutation, which is basically हमने क्या बोला, either it is going to be change in the number or change in the structure, right? तो technical term for change in number is heteroploidy or genomatic mutation. तो इसको हम heteroploidy या genomatic mutation बोलते हैं. And when there is a change in the structure, we call it as either we call it as euploidy, sorry, it is called as chromosomal aberration. तो structure में change chromosomal aberration बोलते हैं. ठीक है. There are two changes in a chromosome. I'm repeating it again. Either it is change in the number or in the structure. Number का term is genomatic mutation or heteroploidy, and structure का change is called chromosomal aberration. ठीक है. Now we are going to make a chart also. So let's keep on making a chart as well. ठीक है. So you've understood this. Anybody who wants to take a picture of this can take, because I'm going to rub this now. Please take a picture if you want to. Now I'm rubbing it. This was just for your understanding, guys. Nobody is going to ask you what is this acrocentric, metacentric. This is just for your understanding, because this is not the part of paper. ये biology का part है. We are not concerned with this, right? So now we have said that chromosomal. Okay. Okay. Let me just write it properly. Okay. इसमें हमने दो बात कर लिया. We talked about heteroploidy and we talked about this is number वाला and structure वाला is chromosomal aberration, which is the number वाला, right? Structure वाला, right? So let's we'll keep on making this chart. Okay, चलो. Now comes let's understand about the changes in the number of chromosome. How many different kind of changes in number can happen? So in there we study euploidy and aneuploidy. So these are two terms under this. So what are the two heads under this? Euploidy and aneuploidy. This is why I am making this chart, to make you understand each and every bit of it. ठीक है? Euploidy and aneuploidy. ठीक है. Now let's go back, look at it and then come back to this picture. Euploidy में क्या होता है? We talked about that we have pairs of chromosomes, so you can call that pairs as sets, right? So you can call that sets also. So when there is a change in the number of sets and there is addition, देखो number में what changes can happen? Either there will be addition of chromosomes or there will be subtraction, right? Number में क्या होगा? या तो increase होंगे या decrease होंगे. That is the only thing that can happen in number. So euploidy में what happens is there is change in the number of sets. Okay. So now let me give you an example. You see this normal karyotype. I am going to show this picture again and again to you, because that is how you are going to understand it. Now assume that this was the actual scenario, and now what happens in each set? One of the, okay, let me draw it only for you. This is the normal karyotype. You get it? This is the normal karyotype. Now let's make a euploidy type of a karyotype. Let me first make the pictures for you. Okay, now see both the pictures. This is one, okay, this is first picture and that's second. This is a normal karyotype wherein we have 23 pair of chromosomes and total 46 pairs. Here in the person, one set, in each set, uh, one part is missing. You can see I have not drawn one part. I have not drawn one part of the pair. You can see that this part that I am drawing with the yellow, I have not drawn. ठीक है? So this is missing. So euploidy में there is this first kind of case wherein in each set, you know, it is not diploid, it is haploid. ठीक है? So this is called euploidy, and in euploidy I have shown you subtraction. First thing I have shown you subtraction. It is called monoploidy. ठीक है? Why is it called monoploidy? Because there should have been diploid cells, but here only monoploid. That means one part has remained. So it has become monoploidy. Now let me show you the next case. ये तो subtraction का case हमने देख लिया. Now let me complete this. Let me complete this karyotype first. Yep. This is Y. Okay. So X. This is X, and this is a Y. I told you X is longer gene. It's a longer as compared to Y. And I told you the reason also, because most of the genes are encoded in the X as compared to the Y chromosome. ठीक है? That is why it is a longer in size. And mostly जो genetic diseases होती है ना, let me not write it here. Mostly जो genetic diseases होती है ना, they are X linked, because most of the genes are encoded on the X. So now I have deviated from the topic, but let's not deviate. So this has now again become a normal karyotype. We are studying euploidy. ठीक है? Now what happens? Just see very carefully. Okay? Now what I have done is, okay, now what can you see? Just look at this. Now in each pair I have shown you that one of the chromosome has been added to it. That means normally there should have been 23 pairs. यहाँ पे भी there are only 23 pairs, but 46 chromosomes should have been there. Now 46 plus 23, each set has added a new chromosome to it. So now what it becomes? It becomes polyploidy. Okay, in polyploidy also, what is it? It's ट्राइप्लॉइडी. ठीक है? इसमें ऐसे ही टेट्राप्लॉइडी हो सकती है. दे कैन बी सो ऑन. टेट्राप्लॉइडी में क्या होगा? वन मोर वुड बी एडेड टू इट. तो ईच सेट विल हैव 4. ठीक है? तो पॉलीप्लॉइडी क्या हो गया? नॉर्मल से मोर देन दैट. नॉर्मल में क्या होना चाहिए था? इन नॉर्मल देयर शुड आइडियली हैव बीन ओनली 2 इन ईच सेट. वेन एवर इट increases from 2 to any other number and every set has that, उसको हम बोलते हैं polyploidy. So now you understand. Let's go back to our chart and add it in that. Okay, first let me see what we were doing. Okay, we have not, अभी कुछ add नहीं करना है इसमें. हमने euploidy, we have understood what is euploidy. Now let's go and understand what is aneuploidy. Aneuploidy में क्या होता है? It is change in the number of chromosome in a set. Now I'll just show you a picture to make you understand. Okay, let's first see. अच्छा, polyploidy तो I explained you ना. In polyploidy, रुको, let me first do the polyploidy only. In polyploidy we have two types, autopolyploidy and allopolyploidy. That means this is just an extra information that you can understand. Polyploidy तो you have understood what it is. Autopolyploidy में क्या होता है? When there is extra set with the same genetic material as the original one, so that becomes auto. Allo is a new extra pair has been added, but with a different genetic material from the original one. ठीक है? So if you have to like show it in a picture, you can say if I have to draw autopolyploidy because it has the same genetic material, so you can say like this. Oh, I'm so sorry. Just wait. Yeah. So if I have to show like autopolyploidy, I can show it like this. ठीक है? So this shows autopolyploidy because same genetic material has been added, and similarly in each pair it is going to be added. And this shows allopolyploidy because different genetic material. So I have changed the color and it signifies the different genetic material. So you understand now. ठीक है? This is auto and allopolyploidy. थोड़ा extra बता दिया. You understand now. Now let's go to the second part, that is aneuploidy. Now what is aneuploidy? Let's look with this picture. Now this is Turner syndrome. Okay, this is Turner syndrome. This is also a you can say this is a genetic disorder. This is a genetic disorder which occurs when there is a change in the number of the chromosomes, and in the sex chromosome there is one chromosome is missing. ठीक है? So you can see it should have been either XX or XY, but it is X and 0. That means एक X, either one X or either one Y is missing in this case. So this is it would have been a normal karyotype had it had this extra this part, but this part is missing. So that is why it has 45 chromosomes instead of 46. So and this disease is called Turner syndrome. When in the genetic or when in the sex pair one chromosome is missing and there is monosomy. So what is this called? This situation is called monosomy. Okay, this is called as, and this is example of aneuploidy. In this is aneuploidy. Euploidy में क्या था? Each pair will either have one or will not have one extra, right? Either every pair will have only one or more than two, either 3, 4 like that, right? तो क्योंकि each pair if has 2 तो normal हो गया, that'll be normal. But if each pair has one or each pair has more than 2, that becomes euploidy. In aneuploidy, only one pair does not have or have extra. So this is one example of aneuploidy. The other example of aneuploidy is Klinefelter syndrome, and there are many others that we are going to study in this chapter. Many genetic disorders we are going to study, so do not worry. इसमें भी what happens, इसमें trisomy है. Like Turner syndrome had monosomy, this has trisomy. Similarly, इसमें भी I have by mistake I have taken the same example. Like I should have taken some other example. So normal karyotype में sex pair में there are two, either XX or XY, but in here it has XXY. So there is one copy extra of X, right? There is one extra copy of X. So this is called trisomy. And so this is aneuploidy. Aneuploidy, again I am telling you what is aneuploidy, wherein not every pair but only one pair has either extra or either has less chromosome, right? So you understand? I hope you do. ठीक है? So this I have told you. So aneuploidy में there are two, hypoaneuploidy and hyperaneuploidy. Hypo क्या हो गया? When there is monosomy or nullisomy. Nullisomy क्या हो गया? Okay, let me show it from my diagram. Let us do पहले, let me rub this and make it a little normal karyotype. Then only I am going to show you, na? Okay. Just wait guys, 1 second. I am just rubbing the extra ones because this will look like euploidy. So let me make normal. Okay. Okay. So for example, if I in the chromosome number 4, I have shown extra that shows trisomy, and that is an example of aneuploidy. This is one kind of example. This is showing trisomy. That means in place of 2 there are 3. This is trisomy. Another example is nullisomy. Okay, so nullisomy के लिए let's do a thing. We rub this. That means 22nd pair is missing. So nullisomy, that means one whole pair is not there. ठीक है? So this is example of nullisomy. Then monosomy की example भी कर लेते हैं. Monosomy, let's do this pair. Okay, so it should have had 2, but in 5 6 7 seventh pair we have only 1. So this is example. These are different type of examples of aneuploidy. इसमें this is hyperaneuploidy, that means extra, and this is hypoaneuploidy because it does not have. And देखो subtraction जहाँ पे होगी that would be hypo, that means this is also hypo, and addition shows hyper, so this is also hyper. So you've understood this. I hope you have understood this. So this happens when there is when the cell division is happening and chromosomes do not separate properly. So that is why it can happen, and it can happen both in the sex and autosomal pairs. That means this numerical aberrations can happen both in the sex as well as in autosome as well as sex pairs. Okay, that means all the 23 pairs it can happen. So I think we'll keep it this much for today because it might become very tough for you. Let's make this tape chart a little bit more. Let's go and add, because हमने इसमें we have understood two types in this. This is one hypoaneuploidy, and then we have studied hyperaneuploidy. ठीक है? In here we studied about monoploidy. Okay, let me write it only. It's monoploidy and polyploidy. ठीक है? Oh, polyploidy. Okay, polyploidy. ठीक है? So I hope you are understanding this table. Tomorrow we'll do it with a lot of many other examples. We have to do chromosomal aberration also, which is related to the structure. Let me just write it here also so that there is no confusion. Okay, so we have to cover a lot of others. Today we have covered the first part, that is in chromosomal mutations. We have studied about the changes in the numerical chromosome number, that is called the heteroploidy, that we have studied. And tomorrow we are going to start with our chromosomal aberration, that is changes in the structure. And uske baad then we are going to do certain famous genetical disorders which happens either in the structure or number of the chromosomes. So guys, if you liked the lecture, please like, share and subscribe. Thank you so much.